Great. We've got some people that are still trickling in, but let's get started. My name is Tim Hunt. I'm the Chief Executive Officer at the Alliance for Regenerative Medicine. And right now, we're going to be talking about cell and gene therapy, ethics, and society. And this is part of a bigger project that we're working on inside of the Alliance for Regenerative Medicine. And for those that were here this morning and heard my opening remarks, I kind of laid out some of the elements of that. And it's sort of born out of what I think of as sort of practical necessity. What do I mean when I say that? You know, we in the community, and certainly we on the ARM team, but we in the broader community, when we're talking to people that we care about, we get asked some basic questions. And when I say people we care about, think about like patients and patient advocates, regulators, payers, right? Whether they're in the United States or in Europe, religious leaders, bioethicists, people, policy leaders, people in our broader community that really want to see the field succeed and really want good things to happen for our patients. They ask some basic questions like, I know this stuff's kind of complex and hard to make, but $2 to $4 million sounds like a lot of money for a medicine. Reasonable question, right? Will only the wealthy have access to these things? Will only the wealthy countries have access? Reasonable question. Is this model sustainable over time, really? Can you keep doing, like, how does that work? How well regulated are all these medicines, right? If you're a patient, you're a parent, and you're going to dose your child, you want your child perhaps to be dosed in a clinical trial, you need to think about it deeply and reflect on it and talk to people about it. So we've assembled a great group of people to talk about what we think of as the ethics within, how cell and gene therapy, ethics, and society are actually harmonious. Or not, they can have different views on this. And what I'd like to do is maybe have everybody go and introduce themselves, and then if you could introduce yourselves, and then reflect on when you think about cell and gene, this topic, cell and gene therapy, ethics and society, what comes to mind for you? What maybe two things come to mind for you? So I'm going to start, Kevin, with you, then we'll go to Matthew, Francesca, and Tay, okay?
Great. Well, thank you so much, Tim, for inviting me here, and thank you all for coming, because I think this is going to be a both interesting and important conversation. So my name is Kevin FitzGerald, as you can see. I'm a Catholic priest. I'm a Jesuit. I'm at Creighton University, which is principally based in Omaha, but we also have a medical center down in Phoenix, and I've been down at that medical center this past year. My own background is I have a Ph.D. in molecular genetics. I've done lab work in this area. I don't know if Lucy is still here, if you heard her story this morning, but I did my postdoc in a lab that worked on a gene at that time was called MLL, mixed lineage leukemia, which is often involved in infant AML and ALL. And I'm just speculating that maybe her son had that MLL translocation, because normally it is a very aggressive tumor and sort of had that profile from what she was describing. But I also have a PhD in bioethics. So I've worked in this intersection of biotechnology, biomedical research, and the larger ethical and social issues for decades. I'm going to date myself now. I was at some of the first conversations that French Anderson raised in the 1980s when he was trying to do SCID gene therapy. And of course, the technology wasn't even close at that time, but at least we were having those conversations. So that's what I think is really important right now. We've been having these conversations for 40 years, but the technology now is at a point where it's really forcing us to make some concrete strategic decisions in how we're going to go forward. And I very much look forward to trying to bring all of that 40 years of conversation to this moment where we can, I think, make some excellent decisions on how not only we can benefit this part of the field, but we can benefit healthcare more broadly. And so that's why I'm excited to be here today. Thank you.
Great. Thanks.
Good afternoon. I'm Matthew Durdy. I'm the Chief Executive of the Cell and Gene Therapy Catapult. I'm a curious mixture from a background point of view. I'm part farmer, biologist by training, economist, and investment banker by first career, and then the last 20 years I've been working in the cell and gene therapy field. The first thing that comes to mind when the word ethics comes up is actually trust. And that's why I think this conversation is so important, because if we are holding the conversation about what we're doing, why we're doing it, and how we are doing it, then we will be able to generate trust from the people that we need to do things. And we need a lot of people to do a lot of things in order to make the industry thrive and work. And that's, so trust is the first word that comes out. In terms of the specifics of ethical issues that then come after that for me, it is about global access. It's about how do we take something that we have been part of creating as an industry that is so powerful and make sure that it's accessible to as many people as possible.
Francesca?
Yes, thank you for the invitation. I am Francesca Pasinelli. I am the CEO of Fondazione Telethon, which is a charity that was born out of the will of rare disease patients more than 30 years ago. And as a charity, we have been funding research throughout all these years. But at a certain point, we started developing gene therapy products. Rare monogenic disorders have been critical in developing gene therapy. I mean, they were the ideal setting for prove the technology. So as soon as we understood that this technology could eventually develop in a treatment, we started building development capacity within the foundation. Given the rarity of these diseases, there was no interest in the pharmaceutical companies at that time. I'm talking 2000, 2004. And once we had a clinical proof of concept, we licensed out our platform for several diseases to a pharmaceutical company. It was GSK at that time, who then out-licensed that to Orchard Therapeutics. And I mean, that was a successful program that ended up with the registration, the market approval of one gene therapy for SCID-ADA, a very rare immunodeficiency, and now to the market approval of metachromatic leukodystrophy. And there are other diseases that are on the clinical development phase. Quite recently, one of these therapies was dismissed by Orchard Therapeutics for commercial reasons, and we decided to become market authorization holders. So we as a charity are, covering now the full path of these diseases. What is gene therapy for us? Gene therapy is a life-saving procedure. What is ethical for us in this process is that we believe there is no rarity that justifies lesser engagement and commitment. And therefore, we believe each single life counts. And that's what we are devoted to. And what we're trying to do is trying to make these programs accessible and sustainable for the rare disease patients that we were born for.
It's amazing work you guys have done in service of patients. Yeah, thank you. Tay,
thanks everyone.
Help us. What do you think about when you hear ethics and cell and gene?
It would be missed if I didn't say the single thing from the outside world usually comes, which is always in my frontal cortex and hits me in the morning, is the sticker shock that comes up, but we pivot away from that quickly when we talk about value. But way of introduction, I head up our Global Value and Access Organization in Novartis Gene Therapies, formerly of Exis. And, it's been a real privilege to not only be participating in the build of and scaling of our proprietary AAV, but I've also had the privileged to participate on our first tech transfer and approval of our CAR-T, which was Kymriah, the story we heard this morning. And again, I heard the story a decade ago of Emily Whitehead, but hearing that story just, it would be hard not to say the two things for me that come to mind. And the first is, and we've sort of converged to this, is firstly, deeply understanding what patients, caregivers, families go through on a daily basis. And why is that important? Because what we learned this morning was it's almost, before we even get to treatment, it's a diagnostic odyssey for them. To then realize there's far or few treatments available. And, we didn't count the beads, that was treatment. But if you go pre the beads, there's probably a whole journey of trying to actually get diagnosed. So that's critical because we're witnessing, I think you mentioned, 14 potential approvals this year. And so this is like, I think, a Star Wars type moment in healthcare systems that were designed for chronic therapies. And it raises the question of, if you can halt a disease or, ensure a child develops within the normal range of development, that could be huge because it could mean that they could sit or walk or swallow or be independent for the rest of their life, which is a huge magnitude. So that's kind of the first thing, which is deeply understanding that journey. And then the second thing is, once you've understood it, it's about democratizing access worldwide. And I'm fortunate I work for a company that's in over 100 countries. And it's an imposed responsibility around how you can then move these therapies, beyond the first world. And I'm not going to say it's straightforward, doing it state by state in the United States, and then commercial plan by plan, or country by country, reimbursement system by reimbursement system in Europe. But it's probably not good enough if we ignore the disproportionate impact, and we're going to talk about sickle cell as well, that exists with these monogenic diseases in other parts of the world. So those were the two things that come to mind.
Great. Good answer. Thank you. Let's talk about an issue that's been in the news a little bit over the last month, which is this European Joint Clinical Assessment. I touched on it earlier today in my opening remarks for the conference. There's a panel on it, I believe it's this afternoon. It's a hot topic, right? And within the JCA, their methodological group, the coordination group, recently came out with their approach, and it heavily discounts the use of single-arm clinical trials as it relates to oncology in general, and then cell and gene therapies. In particular, this is all going to kick into gear starting January 1st, so it's not that far away. But it seems to be pretty problematic. And how do you let everybody and maybe Tay, maybe you can start us, or Matthew, maybe a little bit closer to it, but everybody up here, I think, has a view, because for many clinical trials, I think it's 77% of the approved cell and gene therapies in Europe today were run with single-arm trials, and 54% of the late-stage pipeline is in that. So that could be a large chunk of the patient population that suddenly loses out. And of course, with single-arm trials, you oftentimes can't run double-blind, placebo-controlled trials with an active comparator because they're unethical. So everybody can get a piece of this action a little bit, but maybe Matthew or Tay, if one of you want to start sharing your thoughts.
So before I go deep dive into the actual single-arm trials, I think for those folks who are not deeply engaged in European EMA, joint clinical assessment, HTAs, and then payer negotiations, it's a heavy lift. Yeah. So in the United States, you get an approved full FDA, and then you usually use that for your label population to get coverage. In Europe, you get your EMA label, and then at times, and especially we've learned with our products, you end up getting some form of coverage to the label. In countries like Germany, you get it, but then what ends up happening is you go to every single member state, and you have to then validate the C, which is the clinical assessment. So EMA's already given you a label, and now you're going each country and having, before you jump into a HTA discussion, you're going into a clinical discussion to say, does this data make sense? And look, you know, we've had protracted clinical assessment discussions in a country like France over 3 years, right? Where other countries like Germany, Poland, the UK are already reimbursing. So what that does is it increases a lot of delay. So that's what Tim is referring to. We're at this stage now where you'll get a clinical assessment, you know, hopefully in line with the way the regulatory file will move, and that will then hopefully accelerate access to patients. That's the intent. What's interesting here is I think, and I say this from an industry perspective, we want to make sure we have, you know, the speed. We don't want to delay access. You know, in the field I work in neuroscience, time is neurons for these babies. Secondly, the quality. We want to make sure that in adjacent to whether they're looking at randomized controlled trials or there's going to be a respect and acknowledgement for these technologies and single-arm trials, we want to make sure we weave in indirect treatment comparisons, real-world evidence, because often, you know, when you're going for accelerated approval, the incompleteness of that data is going to then mature over time when you're following patients up. So we follow patients up in Europe for 15 years in our proprietary registry. And then the third is inclusiveness. We want to be part and parcel of all that process as an industry to make sure that you know, there's an understanding of cooperation, convergence, and co-creation. And under specifically the single-arm trials, we know that we learned that in the cell therapy, we went for joint consultation with EMA, and I still have that story where we were at this pivotal moment, lymphoma. We were in a meeting of 20+ regulators from EMA, their staff, and 5 payers. And they were pushing us for, in the meeting, it was a 4-hour meeting for overall survival for lymphoma. And it was only until there was an opportunity to elevate the patient voice, a lymphoma patient, you know, 50+ in the room, in that advice, to say what this company is proposing is progression-free survival for another, you know, between, it was at the time, 12 to 14, and that's huge, 12 to 14 months for this aggressive lymphoma. So we were able to conclude, post that meeting through the write-up, to move away from overall survival to totality of endpoints with a single-arm trial and MRD, negative disease, as a main clinical endpoint. And I look back at hearing this story about single-arm trials that's resurfacing. And if we hadn't agreed that at the time, I think we would have still been waiting in a protracted way to try and get this approved in Europe. So I think as an industry and, you know, as a society and companies and clinicians, patients, I think we as an ecosystem have to use that full force to make sure that, you know, being put in a square peg, in a square hole with small molecules, biologics, with RCTs, with large patient populations, probably is going to push out the timelines for some of this. So they're my initial reactions, but I would want to make sure we push for the efficiency and the speed to make sure we don't then have to repeat all that at member state level, because our goal is to make sure patients can get this accessed as soon as possible.
Yeah, I'm probably old enough, well, I'm old enough that I was taught to do the analysis of RCTs on paper. And I think it's important to recognize that RCTs are brilliant, they're a fantastic construct, and they work really, really well, but they were designed for a different purpose. They were designed for the purpose of decision-making in very large populations with small effects, and enabling also discovery of side effects and the likes within the process of doing that. It's a different environment that we're in here now. And they're an economic measure. They were designed to reduce the amount of data that you had to gather in order to make a decision, where actually our population sizes in clinical trials are quite small. And it seems odd to me that we are still using a mechanism routinely. There are places where it should be applied within cell and gene therapy, but we are doggedly sticking to this. It should be an RCT discussion, when in fact the discussion should be, how do we, rather than use a mechanism that restricts the amount of data that you put into your calculation, why aren't we using mechanisms that absorb as much data as we possibly can? We can do the calculations with computers now. We can gather data much more cheaply than we would have done before. We need to open up a discussion about what is a much better decision-making process associated with small populations where there is a large effect and there is this urgency in terms of getting the product through. It is incumbent upon us as a community to drive that, because the regulators and the medical community are clearly going to sit there and go, RCT, gold standard, we're not going to do anything else. And we're going to have to open their eyes to that, and I think we need to work on it.
Yeah. Francesca?
Yeah, I couldn't agree more, so I won't repeat what was just said. I think a centralized decision is desirable, because, you know, all of these different countries. And so I won't repeat what was just said, because I fully agree. I would like to point out that this is not a European problem only. We very recently had to face a similar request of a bone marrow transplant control arm with gene therapy, which in the phase 1/2 trial was proven to be effective, and ex vivo gene therapy is an autologous transplant, while the bone marrow transplant is an allotransplant that is not, you know, which is a risky procedure that we know. So I'm wondering why the data that we already have regarding the bone marrow transplant couldn't be used as a control arm. So I really have ethical concerns on what it was asked by FDA very recently, and so I think pragmatic, informed decisions on these topics should be undertaken. Very often regulations don't take into account what is really needed case by case, and again, I won't repeat what was said before, which I fully agree.
Thank you. Dr. FitzGerald.
So why does ARM exist? Why is there ARM, right? And it's because together you have a lot more impact than you have individually, right? You come together as an industry to leverage, you know, your goals, to leverage your technology to get the benefits that you want. And I think we need to expand that even more in terms of bringing together all of these sometimes disparate patient advocacy groups. There is an entity in the United States called the Genetic Alliance, which I think now has like 63 different groups together. But I think we can think even more broadly than that and engage the populations around the world and just say, look, we have these opportunities now, which we've been talking about for decades, and it is, as you saw earlier this morning, really now starting to come to fruition. What do we want to do with this? What are your goals, patients? What are your hopes and what are your desires for this technology? And then get that information back to your researchers, back to your regulators, back to your politicians, and help them to understand that these populations become very sophisticated in their understanding of these technologies, the risks of a single-arm trial versus RCT and all this sort of thing, and they can understand and say, yes, we are willing to undertake these risks with you. So we engage them from the beginning, as I know many of your companies have done, in whatever patient advocacy group areas you're involved in. But we've got to now expand that, make that a more global approach in a sense, and get that voice heard so that the focus becomes how do we move these things forward best rather than how do we protect ourselves our political careers, our regulatory careers, our whatever, legal careers, because I think that is currently the focus, when we've sort of lost the idea of the focus being, how do we get the best treatment plans for our patients in play as soon as possible?
Thank you. All right, let's drill into two areas. We'll start, Francesca, with you first on how, just talk maybe a little bit about the Telethon model and how you guys think about access, because it's we could spend the whole hour talking about that component, but it's a very unique kind of case study, and then we'll go to Tay, because the Zolgensma one's very unique from a global access perspective, and he can maybe share some thoughts on sickle cell as well.
Well, first of all, let me tell you that we did decide to develop these therapies in a, let's say, pharmaceutical regulatory standard, according to regulatory standards. So we didn't aim at something which was, less rigid in a way than the other one. Because, again, we believe a rare disease patient deserves and has the same dignity and deserves the same standard and top quality. So either regulations are too strict for the industry and in case we should reduce them, or we have to stick to them. So this is our policy and our position. So, and of course, this is an expensive process for us as a charity as well. So we dedicated a lot of money to that, and partnering with the pharmaceutical industry was a way to minimize these costs, of course, and to accelerate the process. Now, we fully understand that when you talk about ultra-rare diseases, the investment is such, and the cost for maintaining the product of the market, to keep the product on the market, is such that it may not be financially convenient. It may be
commercially viable.
Yeah, worth it. Yeah, sorry, can be worth it. So we decided that we have to do our part. If you think of, and of course we had to adapt ourselves and create the infrastructure to do that. There are a few things that, of course, there are some things that if you look at the cost of the product and the price we are aiming at, we don't have to recover the R&D costs, for example, because all our R&D costs were sustained thanks to donations, and therefore we don't have, we don't need to recover that cost. We will have to negotiate for ultra-rare disorders in particular, but probably that's true for everyone, that some regulatory costs should be reduced. If you keep the product on the market, there are fees that are expensive when you think of products that will treat 3-4 patients a year in Europe, for example. Mm-hmm. And the regulatory costs, the fees, both in each single country where the market is where the product is available, or the EMA cost, will have to be reconsidered if you want to make. And we will try and lobby for that and work for that to see that these are reduced. The other thing is that what we are trying to do is to harmonize, at least for these products, European and the FDA, the EMA and the FDA process. Right. We will probably apply for a market approval for another gene therapy, which we took back. And of course, we won't be able to replicate the clinical studies in the US. This is something we cannot sustain. So our goal is to make the drug available to patients. Then I think each one has to do their own part. and probably looking at dossiers, clinical dossiers, when you talk of monogenic disorders, there is no reason why some clinical trials should be replicated in different areas once you have efficacy data that were also that included non-European patients, including US patients in the first trial. So I think our model will try and is envisaging a path that will take into considerations all these bottlenecks and complications, which we believe are probably not necessary in the access of the product. On the other hand, we are working on optimizing the development through a platform approach, which will try to utilize what we already know of this platform in order to be able to conduct parallel development of more than one gene therapy, minimizing the number of experiments and the time, trying to utilize everything which all the procedures have in common. And we are at present studying a platform approach on 3 diseases that we are conducting. When I say we, I'm talking about the San Raffaele Telethon Institute of Gene Therapy, TGET, which is our research centers where all these therapies have been developed. And this process is being conducted under the continuous discussion with EMA and IFA here in Italy. And that, of course, will eventually and hopefully come to the development of a process that will really minimize time and costs and will allow more diseases to get to a therapy in a much shorter and more convenient times. And also there is a lot that has to be done in optimizing the production, because production is still the most expensive part of this development as far as we are concerned, of course. And that I think we believe will get to again more efficient processes. One thing should be said. There is a need for constant innovation that is increasing the cost of these therapies. Our position is that sometimes marginal benefits in the therapy are not worthy. If you consider that you have technologies, thinking, for example, at lentiviral based gene therapy, is an approach which we know is effective and safe as far as the data we have so far are concerned. And so we believe there is probably no need to try and apply new technology to diseases that already have a therapeutic answer. Again, we are talking about ultra-rare diseases, of course.
Why not move on and go to another disease?
So the way to try and minimize and make it sustainable is looking at it very pragmatically and decide that if you already have a technology which is proven safe and effective, you have to stick to that, because that won't amplify further costs.
Yeah. And then, in other words, there's so many ultra-rare diseases. Move on to the next one and try to help the next patient population.
But some of them are relatively simpler if you are aiming at the proof of concept of a new technology. And therefore you try with that same disease because that accelerates the efficacy of a new technology. Yeah. But again, it increases costs, burden, and then you have to look, you have to ask for a final price of the new therapy, which is probably unsustainable, and again, leave a lot of diseases with the same medical need at the beginning.
Yeah. Good, thank you. Tay, start us off with the Zolgensma case study. A lot of people don't know, I think, that Zolgensma is actually approved and more available in more than 50 markets around the world. it's a very interesting, I think, kind of glimpse of the future of where the field could be going. So maybe touch on that, and then just a minute on the sickle cell work you guys are doing.
Well, yeah, no, happy to. And I want to pick up a theme from the doctor, from Kevin. You've said, why do we kind of exist? Like, what is we get together at the MISA, we get together here, and you find that there's a DNA that people I learned yesterday on a panel I was, learn something, you reset, and then you relearn. And so we were faced with early questions, right? Do we go fast alone? Or do we partner and go? It's gone off. Come off? It's come back on? Yeah, so we were faced with this question day in, day out. Are we going to go fast alone? Or can we go further together? And that was really the principle behind the 50 plus approvals. And I'll start with, Avexis existed in the Midwest, early trials in the Chicago-based plant. And I'm really humbled to say, look, I think Novartis, back to the partnership, I think, making that acquisition allowed us to elevate regulatory thinking, elevate manufacturing capability, and then put the full force of the commercial model behind it. But that again had to be reinvented in real time. So the headlines, we are now approved in 51 markets. We've treated 4,000 plus babies with an AAV. And so you've got to imagine, first pivotal trial, 12 patients treated, and 4,000 in real time about safety, liver tox, collecting data through our registry. We have access now in 5 countries. And that really has been with the principles of 3 Cs of converging the healthcare system around acknowledging and respecting our data, valuing it before they want to jump into a pricing discussion. Secondly, co-creating new solutions, i.e., paying us over 7 years, right? Changing laws in Taiwan and South Korea, changing the way reimbursement's done in Brazil, etc. And the third C is around then how do you cooperate on areas where there's different incentives. And one thing I'm unashamedly really proud of is within 3 years of an US FDA approval, we've been able to take this technology to 7 as of today, low middle income countries, including Brazil, Egypt, Ecuador, etc. And honestly, each time, those beads are still stuck in my head. There were so many ways there were setbacks, or shall we file, shall we not? Will we ever, make it commercially available? But you just somehow have to navigate through that noise and say, with not having all the answers, be comfortable making those recommendations, not having surety of the answer that what's going to come out at the other end. And so I think it's a starting point. The chain of all these approvals and everyone's going. Our hope is that people won't have to repeat that, but almost stand on these models and go further, even more than we've been able to go. So that is the piece. And now we've got data that's 8 years out from the first treated patients, which is phenomenal. Where people are now off a ventilator, they're sitting, walking, gaining milestones in the early childhood that they were supposed to. And you've asked me very briefly to touch on sickle cell disease and why that's dear. Why do we care? I think you outlined very nicely this morning. It stuck with me, the story of one of the patients you met. And, it's not uncommon that you will interact with a sickle cell disease patient, and before you know it, there's a whole ecosystem of patients, families who are affected by this directly, indirectly. And, about 4 years ago, I led a program with the government of Ghana, which we launched in Davos for our CEO at the time. And it wasn't uncommon, Tim, to hear patients and families wanting to end their life. And you deal with all sorts of issues, from tribal, from religious, to newborn screening, to witchcraft, to, what's happened to this child in this village. I mean, these are like heartbreaking stories. We're not even talking about treatment. We're talking about everything else that isn't being spoken about. So what was that? That insight led us to 80% of the disease burden exists outside of the US, in Europe, and disproportionately affects sub-Saharan Africa. So what is the point of developing a gene therapy where people can't get hydroxyurea? Or don't have access to a doctor? I mean, it doesn't make any sense, right? Why would we develop a gene therapy? So we thought at that early stage, let's start, meeting some suppliers around hydroxyurea, pushing the price of the powder down. People ship from Europe, from the US, because they can't get, the markups are too high in sub-Saharan Africa. So that helps you preserve the organs of the children, getting ready for a reality of one day when the Star Wars medicine come, they would have preserved their organs and then they can take it. So we want to build on the great work of the first generation ex vivo gene therapies, but our insights tell us is in these settings, resource constrained, we've got to have a target product profile that's given once directly to the patient in vivo. So that led to this early work led to a partnership with Gates in discovery. We're about, I think, 18 months to 2 years away from the clinic. And embedded within that partnership is to engineer the access, the healthcare system, the follow-up of patients in settings such as sub-Saharan Africa. And so that's our envision, that's what we're hoping to do. And we already know from the beginning that alone we're not going to solve this one, because our early insight already said, we had to work with the World Bank, we had to launch it at Davos, and now organizations like ARM are going to help us elevate and propel that thinking, where other partners will then enable us to develop and take that further together.
Great. Thank you, Tay. I've got a couple more questions, and I want to be sensitive to our time. I'll pick on some special people then. Matthew, we'll start with you. We've talked a little bit about the sort of industrialization and the maturity of the cell and gene therapy field, and how developments in that realm can ultimately, hopefully, pave a better path for more and more global access. And you've given this a lot of thought and spent time on it. Maybe you could share your general thoughts on that, but you have to start with the views that you shared about 10 years ago that you were telling me about. So this is good humor. Buckle up.
I think I can tie these together, because I think the essence of my viewpoint on this is that we are, as an industry, part of a technology cycle, or a series of technology cycles, maybe gene therapy, rare diseases, cell therapy. But the principles are the same. And 12 years ago, as we were setting up cell and gene therapy, I used to sit on a stage like this with people from the biotech and pharma industry and say, cell and gene therapy is coming. And I could hear them chuckles in the audience as people said, and then people said, you know what, you're never going to be able to make it. All right, the regulator's never going to let you put it into people. It won't work anyway. And if you do get it to work, nobody's going to pay you. All right.
The case for optimism by Matthew Durdy.
Absolutely.
Yeah.
And took the wind out of me a few times that. But you look at where we are now. All right, we're over all of that. We're over the proof of concept. But that's the early adopter phase of the development of new technologies. You get a bunch of people who absolutely believe in it, drive it through, make it happen. In that phase, the product and the technology is really, really expensive. You can look at all sorts of other things that fit that bill. So I think now we're at a position in the development of the industry where we're moving into the next phase, where we've got the next group of adopters. It's not necessarily the later adopters yet. But there's another part that goes with this story that more decades ago than I care to remember, I was involved in the rollout of a mobile phone network. And this is in the days when phones were big like this and all the technologies were different. And we were I was on the board, and the management team came to us and they said, we want to rip it all out and start again. And after we started breathing again, we said, why? And they said, well, since we started this rollout, the industry got together and they agreed the basis under which competition takes place, and they agreed the basis under which sharing of technology and collaboration takes place. And that was a document that you're all familiar with now as GSM, the Global Standards Memorandum. And the impact of that was almost immediate in terms of the industry, because what happened was that the manufacturers of the expensive kit, the switch boxes, and to some extent the handsets, basically went, you know what, we only have to produce one type. And they just churned it out. And the cost of those switch boxes reduced by 90% almost overnight. They were all coming out of China and Korea, which meant it was cheaper to finish the job. And it was a better quality outcome because it was connected to the rest of the world. There was an exchange of data. And now we're all completely addicted to these things. They're in our pockets, and they're continuously updating themselves. What was critical in that was the industry got together, and they created their own competition. Right? They were in control of the competition, and they knew what the basis of competition was. And I think now that we're past this proof of concept stage, we need to start thinking that way as an industry. We need to start thinking about what is the basis of competition. And right now, manufacturing is part of the basis of competition. I personally do not believe that in 10 years' time, if we do our job properly, that it will be the basis of competition. I think the basis of competition will be efficacy. And we will have reduced the cost of goods through all of the innovation that's going on so dramatically that we've enabled competition within the therapeutics and between therapeutics, which means in itself the cost of the therapeutics comes down. You get into a virtuous circle because the cost of the therapeutics comes down, uptake goes up, there's more money in the system, there's more profitability in the system because the margins are greater, and you end up with this virtuous circle of reduced cost, higher volume. And that is, for me, the key to access for many, many countries, because you end up with a product that is deliverable at a profit to a much wider community.
Right, and that cycle of, as you've told me on numerous occasions, of higher volume and more competition by itself, once you get there, will continue to just drive an enormous amount of positive outputs.
Yeah, I think this industry has an enormous way to go in terms of its development and its structure. And so it doesn't get lost. A fundamental part of that is interoperability. It's about standards and switching at all levels. Everything that we do, we need to think about, is this cost that we are creating necessary, or can we work together to take that cost out? Whether it's in the hospital system, whether it's in the manufacturing system, whether it's in the public awareness. And the more we do that, the more the overall cost of goods will come down and the volumes will go up, and that's what drives investment.
Great, thank you, fascinating view. Okay, Kevin, over to you on the topic of germline gene editing. We'll jump around the ethics tree a little bit. So I was in Hong Kong back in 2018 when this odious Dr. Hay walked out and said, ta-da, I've just done kind of what had been thought of as crossing really an ethical red line that nobody thought near term, most people hoped, I maybe should say, thought near term would be crossed, and he had just done it. And he had said, well, here we have these new genetically altered children. And we get asked, this topic does come when you talk to bioethicists. Sometimes when you talk to patients, they frequently conflate what is somatic cell gene editing with germline gene editing. So maybe we're in Rome, give us your views, give us the church's views, give us the ethical case on, as you see it, on germline gene editing.
And just to put it in a bit of context too, to show you the relevance of this, I was recently in New Orleans and at Ochsner Health Center talking about the sickle cell. And in the conversation afterwards, one of the doctors said, well, wouldn't it just be better to go in and do the gametes or do the embryos, right? Because now those patients are going to have kids, and those kids are going to have sickle cell, and we're just going to have to do this all over again. Yeah, yes and no. But so as many of you are already aware, the difference physiologically between somatic cell and germline is what cells you're changing, right? What cells you're trying to edit or add a gene or shut down a gene. And so if you're doing something that's going to be heritable, right, sperm and eggs, then that's germline. And the issue there is both quantitative and qualitative in terms of differences. Why? If something goes wrong with your patient, and of course, as we know, these technologies are not perfect by any means, then if you've done somatic cell gene editing, the problem is limited to that patient, right? And so the idea would be, like we do now, even with the two women you heard about this morning who've had gene therapy for sickle cell, they were going to need to be surveilled, right, for many years to come, just to see if the genetic intervention leads to some kind of either unexpected or just undesired outcome. That's, in a sense, manageable. If you do germline, then you're talking about something that not only is going to presumably affect the patient, but all of the patient's offspring. And so the question there then is, what level of efficacy, what level of safety is acceptable? Because now you're talking about a much greater group that is taking this risk, and many of those are future people, right? So you have to take into consideration their potential desires, even though they're not there to express them. In addition, the idea would be that as we become more and more familiar with the epigenetic complications that are going on, what occurs, right, in that process of epigenetic remodeling that goes from generation to generation. And we know now that, even epigenetic effects are inheritable. That adds a whole nother level of complexity. So from the Catholic perspective, and this is actually kind of funny because I was at a meeting with the AAAS, and this was about 2004 or something. We were talking germline gene editing, and of course,
American Academy of
Science. Yeah, well, they go by AAAS now. They gave up on the American thing. And they stood up and they said, we just want you to know, no germline gene editing. We're never going to do it. We're just not there, and it's not going to happen. And I got up and I said, well, we at the Catholic Church are kind of disappointed in, this sort of Neanderthal approach that AAAS is taking, because we're open to germline gene editing as long as, right, it's safe, the benefits far outweigh the burdens, and it doesn't damage the human dignity of the individuals that are being treated. So, of course, that was, kind of a shock. But the idea is, when you stop and think about it, okay, what are your goals? What are you trying to achieve? And will these things somehow contradict the very goals that you say you're trying to achieve? So if you're trying to bring somebody to greater health, improved health, quality of life, and the intervention that you do results in some future harm, diminishment, then you have to take that into consideration as part of that benefits burdens analysis. And so when people talk about, well, yes, but, well, if you're talking future generations, now you have to add much higher levels of potential burden. And we think that's really going to be the challenge with germline. And as you all know, CRISPR, even though it's much better, although we can have that argument whether zinc finger is still more effective and efficient, although maybe harder in upfront costs. But the question is going to be, are we going to be able to develop at some point some editing, some program, some way of doing it, prime editing, base editing, something, or probably going to be a next generation development beyond those. That is going to give us the targeting effectiveness that we are going to require to even start to think about germline. So I think that's where we are now. It's not that we want it to be off the table conceptually, but we want to recognize the fact that germline gene editing is sort of both a quantitative and qualitative step beyond somatic cell. And right now, we're still talking about the risks involved in that. And just one other aspect of that, I think, that we should take into consideration as we go forward. We are all wonderfully responsive to the stories that we hear of the patients who have benefited from these advancing technologies. You know, Lucy this morning talking about Obi, her son, and what CAR-T did for him. But CAR-T doesn't work every time. Do we hear from the patients for whom it doesn't work? What are their responses? Are they saying, that's okay, I knew that going in, it's still worth it? Or are there real regrets? And do they say, if I had only known? Why do we want to know that? Because that's going to help us understand how better we can engage the entire public. Because there will be people for whom these treatments do not work. And we need to take that into consideration and let people know. We understand that. And that doesn't mean if this doesn't work for you, you failed. No, you didn't fail, right? We're still going to care for you. We're still going to be there for you. It's just that we just don't understand completely how we can predict for whom this is going to work and for whom it isn't at this point in time. Yeah. Yeah. And again, think about that for germline. Yeah.
Matthew, please.
I want to ask Kevin a follow-up question to your response on germline editing. So you came up with a technological solution to it, and how do you protect against abuse of that technology? Yeah. You know, what are the societal mechanisms you could put in? I mean, the way we do it at the moment is we just say, no, we're not going to do it.
And I think for right now, that's fair, because that's where we are technologically, right? But that doesn't mean we can't have the conversation. And actually, the conversation, we've been talking about this for like 40 years, when we first, exactly, right? Or you go back all the way to eugenics. So when you talk about how are you going to protect against abuses, who decides what's an abuse? How do you know, right? So we need, and this is nothing new. People have been saying this too for about 10, 15 years. We need to have a global conversation about this. And we talked about before the importance of education, and I do believe that I'm in education, so I should hope I believe that. But that's not going to be enough. There's a whole body of data out there that indicates that you can educate people all you want. That's not necessarily going to make them agree with you. So what you have to be willing to do is engage with these different communities around the world. Maybe in sub-Saharan Africa, they're going to say, you know, we're not really interested in sickle cell gene therapy. If you could just get us hydroxyurea and some other healthcare benefits, that's what we want for now. You got to be ready to hear that, because we cannot say what they should or should not want. We have to find that out. So yes, some education has to be done, but a lot more communication has to be done on what's an abuse, what isn't an abuse. What are we going to be willing to try and not try? I mean, I think we're all in the room aware that germline gene editing is being attempted at various places in the world. Done, of course, rather quietly, because they recognized, and I believe he pronounces it Dr. Ha.
Yeah,
but
sure.
That's how he pronounces his name. Okay, Dr. Ha. He's back, right? He's no longer in prison. He's back. He's active again. So it is moving forward. Wouldn't it benefit all of us if we could say, but we're the ones having the conversation globally. We're not the ones doing this secretly on the side, waiting for it to work so that we can suddenly tout it to the world and become the place where everybody comes for germline gene therapy and we'll make lots of money. Right? That's not our goal primarily. Our goal, as we said, what do the patients need? Well, the only way you can find that out is you got to ask them.
Good way to end this panel. Got to ask the patients. All right, well, join me, please, in thanking Kevin, Matthew, Francesca, and Tay for their great comments.